Hello, friends out there on YouTube. Welcome to Talking Sales. My name's Steve Cross. What a way to spend our Saturday nights, eh? All gathered round the laptop, as has been tradition for centuries, to waiting to find out about these people who think that they've found cells that can talk. Spoiler alert, cells can't talk. My name's Steve. I'm your host for the evening. I am also the person who's clicking all the buttons behind the scenes to make all the people appear and disappear, which means basically if at any point tonight it doesn't seem slick, it's my fault as your host. And if at any point tonight it doesn't seem like you're seeing or hearing the right thing, that's my fault as your host. So anything that goes wrong is entirely me. Welcome to our YouTube. I should let you know that all the way through this, you can leave us comments on YouTube, and we'll be taking questions that you've got for our panelists. And what I can do is I can pull your questions over. I mean, watch. Anything you say on YouTube, I can pull it over into the stream. Everyone can see it, and we'll have it as a question for our incredible panelists. And speaking of panelists, I have no idea what talking cells is. I mean, cells can't talk. So I'm going to call over some of the brains behind this incredible project, and we're going to find out what on earth we're here for. Hello, friends. I'm just going to make us look nice. I might drop the banner temporarily while there's four of us on screen. Frank, Sophie, Alina. In that order, Frank, then Sophie, then Alina. I'd like to know who you are, first of all. Answer that one, and then I'll ask you more questions. Frank, who are you?
Well, surprisingly, I'm Frank Schwach, full name. So I'm a computational biologist, which is almost like a real biologist, just with a computer instead of a pipette. And I work for the Wellcome Sanger Institute. Been there for about 10 years now, a bit more than 10 years actually. And I've been doing quite a lot of public outreach work, which I think we're going to be talking about today.
Amazing. And Sophie, who are you?
Hello, I'm Sophie, obviously. I'm a theatre coach and director. I used to teach A-level drama and theatre studies and creative writing. But now I'm self-employed. I run the Mousehold Theatre Company, which is an outside theatre company for children out here in Norwich. And I'm also an external examiner, and I examine a lot of monologues.
How does one examine a monologue? Is this as in A-level students or degree students performing, or what does?
Yeah. So it's performance units for GCSE and A-level. So I go off to schools within the county and mark them against a mark scheme. But I don't want to panic anyone.
No, I just want to say to teenagers out there from the Norfolk area, watch out. If you see Sophie coming, you'd better be really sharp with the emotion behind your speech. Let's jump over to Alina. Alina, who are you?
Like Frank, I am also at the Genome Campus, Sanger Institute. I've been there for a very long time, long than I care to count. But I am, as opposed to Frank, I am a lab biologist. So I work with the pipette on the bench, and I enjoy it very much. I also have been doing quite a lot of outreach and public engagement for a long time.
So our wonderful visitors out there are they're asking one big question, and that is a question for all of you, and it is what is talking cells? Because cells can't talk. They can't talk, friends. They can't talk.
Well, you say that, but wait until later. You will see a lot of cells talking. So the idea came actually from something else. So Elena and I, we had a project that we called the Theatre of the Cell. So Elena and I have been doing a lot of public outreach together, going to schools, to science festivals and things like that. And this the Theatre of the Cell project, the idea there was that we would take theater approaches into our scientific public outreach. Yeah, and we had this, I think, really lovely project where we used to go into schools and we used to make the children sort of reenact biological processes. So like replicating DNA. DNA, remember, the molecule that stores all the information about living organisms, looks a little bit like a ladder with 4 different nucleobases or different letters, you can imagine them as. So we just used to put different colored baseball caps on the pupils, up to 100 in a hall at one time, and film them from above, make them form the the ladder of the DNA, and then other children would rush through, split them up, and fill it up again to replicate, to copy the DNA. A very very immersive way of experiencing biology and a biological process. And it was great because you had lots of people in close proximity, and it wasn't ideal for the COVID era. So when that happened, we needed something else, and we thought about other ways of doing it. And I was just watching Alan Bennett's Talking Heads one night, and I thought, that's a way of doing it. Because what that does is it uses storytelling instead of enacting things. So in Talking Heads, and I hope you've all seen it, and if not, go and watch it. You're told a story as it happens. You're not seeing the action, but it's storytelling that's used there to tell you what happens, and that's ideal for.
It's what those of us in the industry would call a very cheap way of doing theatre, more than anything else. So, Sophie, can I just jump to you for a second? So you're the theatre brain here. Can you tell us about what we're going to be seeing in dramatic terms?
Yeah. So basically, what you're seeing is, like Frank said, it's storytelling. So everybody has taken an aspect of cellular biology and they've personified that cell. So they found an analogy for the cell, and they've basically made that cell into a human in a human context. So yeah, that's what you're going to see.
Amazing. And Alina, tell me, as you did this entire project, were there any parts where you thought, this is going terribly wrong, this isn't going to work? Were there any moments where it felt shaky?
Not really, no. But all the way through, and particularly before we started, it was really difficult to predict what is going to happen, and whether we will manage to get it going and get people to participate and feel actually confident enough to do it, to develop a story and act it out and film themselves and do it. And I have to say, it is quite amazing. We have fantastic participants, and you will judge that for yourselves.
Fantastic. Well, on that note, we're going to say a temporary goodbye and au revoir, as it were, to Frank, Sophie, and Alina, so that we can start seeing these incredible participants and their incredible videos. So the first person whose video we're going to be seeing is Amy. Hello, Amy. Amy, you're currently muted. Oh, that's intentional. Okay. I just wanted to ask you what your video is called. But should I play the video? You're still muted, I'm afraid, Amy. This happens with all technology stuff. There we go.
Yeah. Confessions of a Cell.
Okay, fantastic. So we are going to have you back straight after we've played this video to have a chat with me and with Rishi, who's also been helping you out. And you're partly here just to cover the time that it takes me to find the right file and click on it.
By Tesco this morning. Can't go on like this, you know. I'm just tired of feeling like there's something wrong with me. People treating me differently, like being blue is lesser. It's probably damaging, you know. It's like I reached my threshold. horrid that I was going to get crushed, so I got up onto the bandstand. And there were so many people around me. It was getting really out of hand. There was a woman shouting. She had a megaphone. She was shouting and someone pushed me. I was so scared. And I fell into the policeman. He's the one that took me here, and I put my hands behind my back. I haven't hurt anyone or anything, I promise. I didn't do anything.
The weather's been really nice today. I think things are getting better. I heard back from the IOPC this morning. They say they're investigating my complaint. They said they have no tolerance of police officers crossing the line, and they'll be assessing the role of discrimination. Feels like things are changing though. Feels like they're finally taking stop and search seriously. Don't think they would have responded before all those protests, not like this. I know things are far from polished, far from pristine, but I keep thinking about what that poet on TV said the other day. She said, we will build, we will reconcile, and we will recover. I hope she's right. She said, there is always light if only we're brave enough to see it. If only we're brave enough to be it.
Amy, what a wonderful, wonderful piece. Very now. And we're now joined by Rishi. So I'm going to get both of you to introduce yourselves and tell our wonderful viewers why you're here. Amy, you obviously made that video, but you've got a day job, haven't you?
Yeah, so I'm a bioinformatician at the EBI, and I'm working on the Human Cell Atlas project, which is a huge international project to map all cell types in the human body.
Fantastic. And Rishi, what brings you to our screen this evening?
Hi there. Yes, I'm Rishi. I'm actually ex-EBI, and now I'm involved in a project called Singing Science, and I have a musical all about genomics. So I think this matches the tying together of art and science.
Fantastic. A lovely microphone in the background to establish your musical credentials. I love it. So, Amy, tell me, it wasn't a video about protests and about policing, was it? Where was the what was the science that we were looking at here? What kind of cell were we trying to understand?
So it wasn't actually specific to a certain cell type, but it was based on inflammation and what happens when cell types or cells become damaged. And they send out these sort of damage associated molecular signals. And inflammation is basically a sort of process where lots of immune cells kind of send signals, and these signals get amplified, it's just kind of like the tissue becomes inflamed, and then it's really important that the immune cells are able to then regulate that response, and if they can regulate it, then the tissue can then repair and recover, and if they can't regulate it appropriately, it can result in many different types of diseases, so one being cancer, metabolism related diseases, inflammatory diseases like inflammatory bowel disease. So yeah, it's kind of like a unifying theme across biology, I guess.
And Rishi, what do you think of Amy's artistic achievement in creating this from just that very sort of cellular thinking to start with?
I think it's been a really good way of bringing together the whole story of the single cell RNA-seq data being analyzed, and then the sort of damage pathway being mapped to a story of someone that's been unfairly treated because they're viewed as damaged in society, and then aligning that to the kind of the sort of protest movements we've seen over the past year was kind of really a novel idea, I think. Yeah,
fantastic piece of work. Brilliant. I'm just looking at my timings. I have to say goodbye to both of you. Thank you so much for introducing that lovely piece of work. Fantastic. So let's say bye to Amy and Rishi. We might be seeing them again. There's lots of people popping back during the evening. And now we're going to fly over to talk to Peter. Peter, hello.
Hello.
Can you tell us all what your video's called while I find it?
So it is called As Rod Sees It. So unlike Amy's, I went for a specific cell the rod cell at the back of our eyes.
Fantastic. Right, let's have a little peek, shall we?
Whether it clouds in the sky, you'll get by. Shh, baby. Give me just 5 more minutes to have my coffee and peace. 2 shots and 1.5 teaspoons of sugar. Perfect. She'll doze now, just as long as I reassure her I'm here from time to time by singing her song. I like this part of the day. It's when you feel like there's no one else awake and you've got the whole world to yourself. It's when I feel most alive. It's the time for checking that I've got everything that I need ready for the day, ready for every eventuality. God knows there won't be time later to be standing around having the time to think. Na 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 na. Shh. I'm lucky that she sleeps right through. Always has done. It's all about the food, you know, and the routine. I keep to my routine every day without fail. Every morning the same. A few minutes before dawn, get up, get started. My baby has everything she needs when it's right for her. And we've got a good routine with a good structure. She's getting on so well with her stabilisers. She knows that she's looked after and protected. She's got everything that she could want. Who knows what's going on inside that brain of hers, but however it seems to her, it's not so bad. She's got me looking after her. Na 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 na. Shh. Won't be long now. Any moment. And the first beams of sunlight will cross her eyelids, and she'll be wide awake and clamouring for her breakfast. That generally keeps her calm for a bit. Na 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 na. Light up your face now, now, now. Shh. 4 hard-boiled eggs for breakfast. Free range, of course. Got them ready last night. 4 might sound a lot, but she only eats the yolks. I've tried to get her to eat the whites. The protein would be good for her, but she'll have none of it. So instead, it's me that's got to eat them. Even with a little bit of salt. They don't taste as much. But we're on a limited budget, and I'm not letting good food go to waste. No one ever tells you how hard it is to be responsible for the happiness, the existence of another life. There's no manual, and however similar we all look, we're all different. I used to really worry that I was getting it right or wrong, but then I realised whatever I do doesn't make the slightest bit of difference. It's all about the phase she's in. I have to say, this year has been really tough. Doesn't matter, she'll be up in a moment in any case. I can go for days without having another normal adult conversation. I miss my wife, who she was. We were so in love, still are. I'm religious. For richer, for poorer, in sickness and in health, till death do us part. I'm coming, baby. That's it for the day now. She'll be full on the rest of the day. Even in her currently limited capacity, she's got quite the dramatic flair. Had the most awful tantrum yesterday when the tiniest thing upset her. I'm coming. That's the time you must keep on trying. Smile, what's the use of crying? You'll find that life is still worthwhile. No, just smile. Shh. With hindsight, today was a good one. Couldn't be further removed from yesterday, though I suppose that was kind of fun in its own way too. She must be in a mixed episode at the moment. We talked about the summer, how she's looking forward to seeing people in person again. I had to point out that we're not in a position to plan a 40th birthday at this stage. Na 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 na. Shh. Suppose it's fair to say that my love for her has changed over the years. But isn't that true of every relationship over time? And whoever we become, we'll always have a sense of that connection deep within. That connection. We feel it in different ways at different times. For me, it's the sense of the chemistry between us that mean that however disastrous and frustrating she's being, I'll always want to reach out and protect her in the lows and set her free to fly in the highs. For her, I think it's more the sparks of hopes and dreams that our relationship signifies. Especially when she's in a manic phase. It's not just need. Na 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 na. Shh. Of course, it's challenging. With the continual fears and not knowing and questioning what's real. Most people don't have to think about that. Even the normal days, what are they leading to? But we have to celebrate that this is part of the background of who she is, who we are. To think that I thought I could choose my own career. Na 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 na. Shh. Nearly 20 years, and I wouldn't change a thing, even if I could. She makes an adventure out of every day. She's what I was brought into this world. It's true that with her, I don't have a life of my own. But without her, I would cease to exist.
If you smile through your fear and sorrow, smile and maybe tomorrow you'll see the sun come shining through.
How many eggs did you eat doing that?
I've still got eggs for lunch for quite a few days to go, as long as they don't go mouldy.
Amazing. And welcome also James, who's coming to join us to chat about Peter's film. Before we find out who you are, James, we're going to find out who Peter is. Peter, so when you're not making too many eggs, what do you do with your life?
I'm a biology teacher and chaplain at Kimbolton School.
Wow, amazing. And James, how about you? What do you do?
So I'm a bioinformatician at EBI in Hingston. I deal with computer databases about chemistry and biology. So I haven't sort of touched this thing in years and have been dragging out my undergraduate textbooks to remember what a rod cell is. I don't recall them singing.
Well, no, this is all the questions we've got to ask. James, so is this the first time you've experienced this video, James?
I had a look at it just before the stream to get some idea of what Peter was going to say of this. But
well,
yeah,
what questions? What questions do you have for the mind behind this piece?
Well, there's something. So, what made you want to do a rod cell? Why not a cone cell or the other thing on back in the?
Well, it's not just in the undergraduate biology syllabus, it's also in the A-level syllabus, and it comes fairly near the end in the course that I'm teaching, when students are trying to get their head around the brain and what a neuron does, and then they finally got their head around synapses and the connections between nerve cells, and then the rod cell is introduced as the sort of pièce de résistance that they've got to understand. And it's particularly complicated because when the rod cell's active and releasing its neurotransmitter, that suppresses the bipolar cell that's next to it and stops it sending a signal to the brain.
So incredible. I never realized the rod cell was the cherry on the Bakewell tart of A-level biology before. So, I mean, Peter, if I had to summarize this video to someone who'd never seen it, I would just be saying humming and eggs. So which would you like to explain first? Do you want to explain the humming or do you want to explain the eggs?
Let's go for the humming.
Good, right.
So I had
Singing,
to learn how to sing inwards, which anyone watching can try at home, because it is possible. But there were the NAs going out. So Na is sodium, Na. And so that's leaving the cell all the time.
Right.
And sodium's got a positive charge. Sodium ion's got a positive charge. And in the dark, channels are open so that the sodium can come in as well. And I was trying to work out how to do this. I'd figured out quite early on that I was going to have na, na, na singing along to a song for the sodium going out, but I couldn't work out how to get it in. And I suddenly realized you can actually sing backwards. I'm not sure it's too good for your vocal cords, but I managed.
Can you show us what you mean? Because I didn't pick that up on the video.
So I'll go out first and then in. Na 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 na. So it's a sort of yawny bit.
So when you sound slightly like a frog with a bunged up nose, that's you breathing in.
Yes, absolutely. Maybe that's what frogs are doing.
Incredible. And James, so I just wanted to, if you don't mind, I'm just going to ask James a quick question. James, now that you've seen it, what have you taken away from it?
It was not, in fact, about monks doing Gregorian chants like I thought in the first 30 seconds of a dressing gown. I was taken away for looking at that, thinking, what's going on at the back of my eyeballs? Back of my eyeballs, I've got a bit of a creepy codependent relationship going on there.
It didn't seem creepy codependent. It seemed lovely.
A lot of the talking heads videos, there's a sort of slightly negative undertone to quite a few of the relationships that are described in them. So I wasn't sure how to take that, but I wanted to end on a positive note, and it's in the dark, so I thought I'd flip it around.
It was
And
very
Peter,
meta.
the she you're talking about, is that a metaphorical, like a cone cell? Is it the person who the rod cell is in? Is it just, in fact, transcending the cellular metaphor and talking about human life?
So on the biological level, there is a cell called the bipolar cell, which the rod cell connects to. And that's the cell that's being suppressed. So every time there's a that's the suppression by the neurotransmitter that happens in the dark. But then in the daylight, and I tried to show this with the fridge opening and the light from the fridge, but of course, with the wonders of iPhones, the light balance was fine for the dark as well. But anyway, with the light, those eggs, the egg white, which represents the protein, as rhodopsin gets broken down, and opsin, the protein part, is heavily involved in blocking the sodium channels so that the cell can no longer take in the positivity and it becomes negative and it stops shushing the bipolar cell. And I wanted to make it ambiguous in the script in a way that a lot of the talking heads videos go sort of, hopefully you're not sure at the beginning, or you think maybe it's actually his baby that he's raising on his own as a single parent. And then you realize, oh no, this is actually a relationship that's been going on for 20 years. And she's discussing 40th birthdays.
Wow. Well, congratulations on making it. James, thank you for joining us to chat about it. I'm going to send you both the way back into the digital ether, because I need to summon up our next incredible video maker, who is Kaya. Let's bring Kaya up. Kaya, hello. How are you?
Hello, I'm good, thank you. And you?
And would you like to tell us the title of your video?
Yes, it's called Dear Yours.
All right, let's have a look.
I received a letter today. Quite peculiar, I'd say. I'm not that different from anyone else round here, you see, but this letter made me feel special. Dear Anne, I've heard all about you and your beauty. Your smile can brighten up the gloomiest of days. Your eyes can guide the wanderer through the darkest of mazes. Your voice can soften the toughest of men. I can't wait to meet you very soon, my dear. Your B.A. It made me blush. Such kind and flattering words, so much passion in them. It was quite intimidating, but in the good sense of the word, of course. There's just one thing. I'm not really sure who that mysterious B.A. could be. Oh well, never mind. I must know who that sweetheart is. It will eventually come to mind. I can't wait to meet him too. It's a lovely day today. A perfect one for meeting a gentleman. I went around my neighborhood this morning, as usual. I ran errands, sent many packages, exchanged greetings and vigorous handshakes, and stopped at a few houses. For gossip. How could I even function without such a good and effective way of communication? No news can escape from me. Bit of a busy morning there. I'd like to rest, but there's constantly something to do. I've already drank 2 coffees today, and I can feel the energy kicking in, a tingling sensation that helps me be more positive to the negativity around me. at least for some time. I better make good use of it while it lasts. I can't wait to see what the future brings. I've always been told there is so much potential in me, and I am ready to take action, whether in life or in love. Oh, another letter came in today. More letters arrived. It would be quite nice to know somebody cares so deeply about me, if it weren't for the fact there were 20 of them. All at once. I was supposed to meet BA today, and I thought that the letters would give me some directions about where to go and what to expect, or perhaps not to expect anything in case BA had changed his mind. But no, there was nothing of the sort to be found in them. They were sweet and genuine, I have to say, and I've read each of them very carefully, looking for clues. But I can't seem to understand. Oh, there was one thing that caught my eye. He finally mentioned he learned about my beauty and fascinating nature from a nobleman who goes by the initials APP. He apparently had a secret ace up his sleeve, but that hasn't helped much. I'm starting to worry a bit. But on the other hand, I've forgotten what it feels like to be in love. It's been so long. Maybe this is what affection looks like. BA must be a very caring man. They keep coming. It began with one letter, then 2, 3, 40, 70. There are thousands of them now. Each of them separately is just a lovely note in a small envelope. But together, they form a monstrous mountain that weighs me down more and more every single day. It's like they've stuck to the walls of my house, the air around, each other, to me. BA is inscribed deep into my soul, and I thought he would be the one. But he was just taking his time and preparing to slowly kill the N I once was. I stopped answering the doorbell. My door is locked, nobody can reach me. All my connections are lost. I'm all alone. The letters have poisoned me, intoxicated with no escape. And I'm sure I've been sending them back, but they keep finding their way back to me, and I can't do anything about it. They are a barrier between me and the outside world that keeps growing higher and higher until he gets what he wants. Me. The last time I saw my neighbor, she was so excited. The happiest I've ever seen her. And as I wait here, I remember her wide smile, her glistening eyes, and her soothing voice, as she exclaimed the words that haunt me to this very moment. I received a letter today.
Kaya, Birgit, hello. Kaya, incredible piece. Let's find out who everybody is, and then we're going to pick your brains about the mysterious BA. So, Kaya, tell me, who are you?
I'm in my last year of A-levels, studying biology, chemistry, psychology, and drama. And I am preparing to study neuroscience at university.
Amazing, amazing. And Birgit, who are you?
I also work at the EBI, and I'm a curator, which means I deal with the data that's coming in, and in particular, I deal with the protein complexes. So when I heard about Kaya's project, it was like, yeah, I know about this because I actually dealt with the topic in my work. So, but I will let Kaya explain what the mysterious BA is.
Yeah, Kaya. So we've got a protein complex expert here. It's time to spill the beans.
Well, my cell was actually a neuron cell that was slowly being affected by the Alzheimer's disease, and hence the BA is the beta amyloid protein, or amyloid beta, which forms the plaques that necessarily kill the neuron at the end, together with the tau tangles that I didn't show in my video, but I focused on the beta amyloid protein. And also the mysterious APP was the amyloid precursor protein from which the beta amyloid protein is derived. So it was all like a mystery.
Fantastic. And Birgit, so you're our protein sticking together expert. Watching Kaya's film, how did you feel about the way it represented this particular example of the science that you're into?
Yeah, I thought it was really, really beautifully made, and I would like to know which little analogy is your favorite one, because there are obviously lots of little subtle hints about both what the cell is going through, but also what the patient is going through, about how the disease is progressing, and how it's quite disturbing for a person with dementia. So yeah, what was your favorite bit about that?
Well, I loved making it, but I think that something that I hadn't thought before, and then it suddenly sparked into my mind during the process, is at the beginning I say that I've just drank 2 coffees and I can feel the energy that helps me be more positive to the negativity around me. So when an action potential is sent along the neuron, the inside becomes more positive than the outside. So usually the inside is more negative than the outside, but during that time it's the other way around. And then when I said that, well, at least for some time, because the action potentials go and then they come back again. So I think that was one that was kind of not expected before making it.
Yeah, so that's really interesting. I'm not going to give much away about the panelists. Also, most of us made some videos which will be available later. I think someone's going to put a link in the chat. So all of these videos will be available later once they have been edited. And it's true, once you start writing it, there's just so much that comes to mind. And how did you find actually fitting it all in? So you said you've got this disjoined passion for biology and neuroscience. So how do you combine this all? And how are you going to carry on doing drama when you elbow deep in lab work at university?
Well, for the longest time, I had no idea how I could combine them. But thanks to such initiatives, I now have the idea. And it kind of all started with a project that I did for school, investigating the effect of performing arts on our perception of mental illnesses. So I created a song, and I fortunately managed to change the attitude to mental illnesses among my community. And I would love to continue doing that in the future, and to help people with mental illnesses and various diseases come to terms with their disease and destigmatize it, because there's nothing wrong with that. And we should all know that. So that's
fantastic. What a wonderfully positive note to finish this conversation for now on. I'm sure the two of you will be talking for many years to come about all sorts of things. So thank you so much, Karen Birgit. I'm going to send you back into the cloud. That's where you get stored when all of this happens, just stored in the cloud. And I'm going to bring over Rebecca. Hello, Rebecca.
Hello.
Now, can you tell us the name of your film just before I bring it up to show it to everybody?
Yes, it's Alpha, Beta, Gamma.
All right, here we go.
I'm here, first day in the new job. They told me I'd done right well in alpha globin, so they promoted me to supervisor of beta globin. I've had 23 coffees already this morning. At least the canteen's well stocked. I'm kind of nervous. I've heard some things about beta globin, but they can't be true, right? Probably just my old pals in alpha globin joking with me. There's always been this big rivalry over who completes the orders the fastest. It's silly, really. Just like the boss says, we might be making different subunits, but they're all part of the same hemoglobin product. It's not looking good. I had a meeting with the boss. She said we've been having a lot of complaints about our product, and if it doesn't improve, the customer will end up doing an import of red blood cells again. I couldn't believe it. I'd heard rumours of imports, but up until now I didn't know they really happened. Al, my old supervisor from Alpha Globin, was outraged. He said there's nothing wrong with our hemoglobin, and the boss said, well, there jolly well must be something wrong if we're getting complaints. Al said there's nothing wrong with the alpha chains my team make. Then he looked at me. You, he said, it must be your fault. There's got to be something wrong with the beta chains. And I said, how dare you make such an accusation? Well, that's what I would have said if the boss hadn't have interrupted and yelled that we'd both be fired if we didn't find the problem quick. On the way back down the corridor, I said to Al, well, what's the worst that can happen? Is the world gonna end if the hemoglobin's got a few mistakes? And he looked at me and said, listen, we've all heard of that cell that made, I cut him off, too many mistakes and committed apoptosis. Yeah, we've all heard that story 1,000,000 times. Well, I've been checking all day. I can't find any mistakes. No more than the usual anyway. Matches the messenger RNA just fine. Something weird's going on here. Well, it happened. The customer has only gone and put in an order for some new red blood cells without even consulting us. I know the customer's always right, but we hardly got a chance to investigate properly, did we? Al said he didn't find any problems with the alpha product, and then he looked at me accusingly. I thought he was my mate. But no matter what he might think, there's nothing wrong with the beta globin. Not that I can see anyway. So I had a word with my team. None of them seemed to have a clue, other than the longest serving member. She said, I've always said our factory wasn't set up right. Something was wrong with the blueprints. Back in the old days, I used to work on gamma globin line. Never had no complaints about the alpha gamma hemoglobin. I shook my head. But the gamma globin line's been decommissioned for years now. The old timer tutted. Should have kept it going, but no one listens to me. So you know what I did? I got on the phone to HQ. After an hour of listening to the same song going round and round on hold, I finally got through to one of the transcription interns. But as soon as I mentioned gamma globin, he hung up. Something fishy's going on here. I'm gonna find out what. He called me back. That intern from HQ started wittering about people in his department going missing. And I said, well, that's weird, but what's it got to do with us? Then he said he'd heard something about crisps. And I was like, what are you talking about, man? And then he said he had to go. Anyway, I thought, that's an idea. I quite fancy some crisps. But now I think about it, I hadn't got around to properly telling him about my idea to reopen the gamma globin line. You know, make a product that actually works. I thought people were supposed to listen to you when you're in management. You never guess what? I got here as normal this morning, and they've only gone and decommissioned the beta globin line. My heart sank. I thought I was a goner. I was duly gonna be fired, or worse. But they put me in charge of the newly reopened gamma globin line. The old timer kept on repeating, "Told you so," whenever I was in earshot. And then I realised that phone call to HQ must have worked. It must have just sounded like they weren't interested. Yeah, just pretending. They work in some mysterious ways up there. But I did it. I told you I'd turn this place around. It was all thanks to my hard work. Yep, that's what it was. The boss is really happy with us for once. Says the customer won't need to do any more of them expensive imports of red blood cells from outside again. Isn't that great? I popped in to see my old pals at the alpha globin factory. Al wandered over, looking pretty smug. "Told you so," he said. "Told you there was something wrong with your line." And it was thanks to me that the problem was found, I told him. Al looked at me and asked, "Well, what was the problem?" Good question. Well, I've been reading a book on management, so I said to him, "That's a trade secret." What was the problem though? How did they know to reopen the gamma line? Must have been reading my mind.
Wow. All the video makers tonight just on a quest to make me hungry. We've got Birgit back, our protein sticking together expert. Rebecca, tell us, what do you do when you're not pretending to be a factory assembly line boss globin person?
Well, I'm currently studying a master's degree in molecular biology, but I did actually used to work in manufacturing for a number of years, and that was actually where I got my props and costumes from.
Just left over from the old days.
Yeah.
Phenomenal, phenomenal. So I'm going to go straight in because I'm a great lover of snacks. Crisps, tell me, because I was following all of that. I was like blood, and then crisps, and then all I thought about after that was crisps. So why have you got crisps in your video?
Well, it's not actually crisps. The supervisor had actually misheard. The intern was actually speaking about CRISPR, which the first time I heard it, I must admit, I thought that sounds very like crisps. So that's where the idea for that came from. And CRISPR stands for clustered regularly interspersed short palindromic repeats. So it's a little bit of a mouthful.
Come on, no one else on this entire call knew that. Brilliant. Birgit, how do you feel having seen this wonderful piece where Rebecca pulled, like she pulled back things from her past life and brought them into her current life?
I'm loving it. And for me, hemoglobin is always going to be really close to my heart, because when I started this job, it was the first mammalian complex I worked on. And it's just so fascinating because there isn't just gamma, alpha, beta, and gamma. there's epsilon and zeta, and they make all sorts of different combinations. And I don't know, Rebecca, what you wanted to say about how amazing our bodies are to know exactly which gene to switch on to make the right hemoglobin combination at the right time in the in development.
Yeah, it's quite amazing, and just as amazing, CRISPR therapy can now free people with blood disorders from a life of blood transfusions. It can actually reverse the hemoglobin back to the fetal version, which is made out of alpha and gamma globin instead of alpha and beta globin, because in some blood disorders like beta thalassemia, there's a problem with the beta globin protein, part of the hemoglobin, and so if we just get rid of that and get gamma back turned on, and you can have alpha gamma hemoglobin, which works perfectly fine.
Rebecca, do you have a cheat sheet for all of these science facts in front of you? Or is this actually deep knowledge? Because somebody needs to give you that master's degree as fast as possible if you can just reel off globin facts like this. I'm sorry to both of you, but I have to finish this conversation here so that I can fly to our next video. I know we've all got a million more things to think about, but you'll just have to accept all the praise that you're getting in the YouTube chat in lieu of more time to chat. So thanks very much, Rebecca and Birgit, and I'm going to bring out, I'm going to reach into the cloud and I'm going to pull out our next video maker, who's Alina from earlier, who we met. Hello, Alina. How are things? How have you been up to anything good since the beginning of this?
Yeah, hard work as every day.
Brilliant.
Yeah.
So, and tell me, what's your video called that we're about to watch?
It is a tumultuous affair.
All right, let's have a look.
I had a really pleasant round today. Everyone was outside, smiling, greeting, little chats here and there. Very nice. I did notice something though. Little things like a damaged flower bed, broken gate, a snapped branch, a hole in the hedge. Nothing major, but I don't know. Something is amiss. I was just doing my evening rounds now, and on the high street I saw something past me, like a shadow or something that leapt into the dark alley. I followed suit, but I lost them in the dark. I stopped, I looked around, everything seemed quiet, but I did catch a glimpse of a light in one of the houses. So I went over to check it out, and sure enough, there was a light moving around inside. I thought I'd get in through the back door and catch them, but as I was getting there, I saw another shadow running past me. So I hid in the bushes, and I saw another one, and another one, and another one. There were so many of them. I took out my canister of nitric oxide and sprayed them all, and now I need to call for reinforcements. Hello? Lymphoid Corps HQ. Macrophage here. Intrusion confirmed. We need reinforcements straight away. Yeah. Send B and T divisions right away. Yeah. Uh-huh. Understood. Gotta go. What a night. I had to come back for another canister of nitric oxide. Not nitrous oxide, mind. If I wanted them laughing, I would have tickled them. Anyway, the T division arrived soon enough, with the cytotoxic brigade setting straight away to pursuing the intruders to taser them and inactivate them. In the meantime, the helper brigade from the T division spread around to gather information about the intruders and pass it on to everyone else so that we know the numbers and resources that we need to confront the intrusion. Not long after, the B division arrived, straight away catching up with the intruders and handcuffing them. The handcuffs are very useful for us because they give us a handle to hold on to the intruders so that we can put them away. The NK division, that's from the lymphoid corps, they did a sweep of all the buildings and houses just to make sure that there aren't any intruders hiding away in there. And finally, the APC brigade, and that's from my division, from the myeloid corps, they recover all the debris and remains to gather information about the intruders and then pass it on to everyone else so that we know who is who and so that we know who are we dealing with. We also take pictures to keep a record that might come handy later on. But for now, we overpower them, that's for sure. This is the end of this intrusion. It's so reassuring to know that we have such an efficient and well-organized field army. Good morning. I am on the news bulletin today to reassure you about the events of last night that I am certain you're aware of. Myself and my colleagues identified signs of an intrusion yesterday that was confirmed in the early hours of the evening. The identified antisocial elements caused havoc around town and are responsible for the damage that you might have seen or experienced. Our forces quickly deployed a complex operation involving the B and T divisions, as well as APC intelligence and NK surveillance. I am able to report that all the intruders were identified, apprehended, and put away, with some loss of life, mainly on the side of the intruders. We have taken pictures to facilitate swift identification should similar individuals come our way again. But for now, danger has passed and we can start concentrating in repairing the damage. We thank you for your cooperation as we return to normal. Have a good day.
Alina, before I bring back all of our wonderful colleagues, I just want to know this, the camo gear, is that from your previous job? Same as Rebecca's high-vis was?
No, that
Okay,
is
so you're not...
No, that is a costume I borrowed from my 11-year-old child.
Right, let's bring everybody back to have a chat about this. Let's get our full
Who was the
look,
one?
'cause
Who
I'm
was
not...
the one who edited the video, by the way?
Wow, this is why we all need 11-year-olds, like young teenagers around. They can do all this sort of stuff for us. You just throw an iPad at them and you go, make me a Hollywood movie, and it'll be great. So we've seen all of the videos now, team, and I wanted to ask all of you, you've done your very, very intense week of supporting people. They've created all these videos. I just wanted to ask each of you in turn, like, what are you thinking about now? How are you feeling about the project? What does it feel like to have done this, you know? And we've just seen Alina's video, so I'm going to start with Alina because I know she's in a talkative mood. Alina, tell me, you finished all of this for now. How are you?
I am ecstatic, I have to say, because it worked so well, and it was a lot of work, but I think it was well worth it. And actually, I just can't wait to do the next one.
Phenomenal. And Sophie, I mean, I've worked on a lot of projects with a lot of scientists. Presumably you've held a lot of hands throughout this entire process. How are you feeling about the state that it's in now that it's this wonderful set of videos with this very creative set of people? Also, before you answer, you are currently muted. I just thought I'd mention that before you start telling me things. Don't worry. Every single... Oh, Sophie's gone. So we're gonna talk to Sophie again in a second, but instead we're just going to pop over to Frank for a second. Hello, Frank. It's gone dark behind you. That's a nice... I feel like there's been some development there. This is a good piece. How do you feel now that all of this, the twinkle that some of you had in your eye, is now this incredible piece?
I'm delighted and still blown away by the quality of what our participants have produced here. It surpasses my wildest dreams, I have to say. There's so much talent out there. It's amazing.
Brilliant. Let's jump back to Sophie. Sophie, you've held a lot of hands. You've taught a lot of people. Imagine you're examining these monologues now. How do you feel about the project at this point?
Well, yeah, exactly like Frank said, I've also been completely bowled over by the quality. When I first came into this project, I thought, okay, science and drama, how's this going to work? I mean, they're two completely different fields, and there's not very many people like Kaya who, and all of the other participants actually, who are very good at joining and fusing those two things together. So it started off with me delivering 2 hour-and-a-half workshops on how to write, act, and film the monologues. And for me, from the get-go, I was kind of like, okay, so I've got to teach them a kind of lifelong amount of work in 3 hours. And then on top of that, obviously, they've got to have the biology knowledge or do the research on the biology. And I just think it's incredible what everybody has achieved, including panel members who have also made videos. Because not only have they had to do those things, they've had to write the script, make up their analogy between the cell and the person that they're playing or the character, learn their lines, film it, edit it. You know, they've had to you'd have to do the set, the costumes, be the art director, and then sort out the footage. And some of the participants actually edited their own videos when they didn't have those editing skills. So they've done well on that count as well. And then I've edited some of the other videos. So I've had to work with the participants on that. So, I mean, there's a whole lot of things going on here. And I do feel like I need to watch all of these videos about another 10 times each before I even get half the meanings that they've embedded within them.
Incredible. And Rishi, you've worked alongside everybody through the whole project, guiding them based on your many years of trying to bring together science and very disparate creative forms. How are you feeling at this point?
I think a sense of wonder, in particular at, us panel members, as Sophie pointed out, have had a go at making these videos, and you sort of took a month or 2 months to produce something, and they've taken on board what Sophie presented in her workshop and produced this. The Cambridge Festival was just a week and a half ago, and they've taken on board the knowledge she's imparted, researched the biology, and produced a whole range of a variety of wonderful stories, just showing how wonderful the body is with so many body parts like eye cells and brain cells, and that variety and the quality of what's come up in such a short amount of time. I have to take my hat off to them. In fact, I already have.
And a question for all of you. So you've done so much, but it feels to me like you've opened a whole load of doors, and there's a whole lot of things behind those doors to explore. So what's this project, and what is this team and this way of thinking going to do next?
Yeah, that's a really good question. I think we've got lots of plans. There's a lot we can do with this. I think the best thing about this is that we don't use computer animation or things like that. There's lots of YouTube videos that show biological processes with these sort of means. That's really hard to do. We just use storytelling, and we're all storytellers, aren't we? All we need is a campfire somewhere, get around it and tell a story. And so this can be done anywhere. Yeah, you can. Everybody has a camera nowadays, so film yourself, do it wherever you want. So we've got lots of plans. One of the plans is that we will try to work with patients, with people who are affected by certain conditions, almost as a sort of therapeutic thing to engage with the science behind something that affects you so deeply. And I think this sort of theatrical approach would work quite well with it. And also, I think we should develop something that's more aimed at younger audience, at children, to do at schools. We've deliberately kept this one as a sort of for adults, really, because we just wanted to do something like that. But there's lots of ways we can explore here.
Sophie, you're going to be working with this lot again?
I hope so. They're a fantastic team, and I've kind of had to deal with my own insecurities in that I just simply do not know as much as them by any stretch of the imagination.
Really? I mean, how many times did you stay past midnight working on this? I thought you would be glad to see the back of us.
This week it was 3 a.m. I regularly saw. Yeah.
Sorry.
Well,
whatever they're paying you, Sophie, it should be more. If you're out there, Wellcome Genome funding people, just pay Sophie more. I'd say that as freelancer to freelancer, you know. What a wonderful thing. Congratulations to all of you. Congratulations to everybody else that we've met through this lovely evening. It's been such a pleasure hanging out with you. Thank you all out there in YouTube land for joining us. It's been a real pleasure talking to you all. These videos are all available through the Wellcome Genome Campus Connecting Science YouTube channel. That's youtube.com/Wellcome Genome Campus Connecting Science YouTube channel.html. It's not, I don't know what it is. You'll find it without too many problems. You'll be able to see all these videos. Sorry, Rishi?
We've added links into the chat.
You've added real links. Okay. No one has to type in the whole of youtube.com/Wellcome Genome Campus Connecting Science YouTube channel all into one long thing. It's been a real pleasure. I'm going to let all of this core team wave goodbye to you all, and you'll all have to go back to your real lives, and we'll never really find out who got the crisps. Goodbye, everybody. Have a brilliant evening.
Cheers.
Bye-bye.